So if you have like privacy, you can just turn off your camera.
I don't know what I did, but I lost my screen. 1 second. Okay, I'm back again.
Okay, just hola a todos y todas. Hello, everyone. We are happy to have you here to enjoy with us one more session of our monthly Cactaceae Seminars. Our initiative with the Cactaceae Seminars is trying to keep our community engaged with the research and the outreach topics related to cacti. So just as an announcement, if you are on Twitter, we now have a profile, so you can find as Cactaceae Seminars, and you can help following us and sharing the next seminars. Today we are welcoming Mónica Arakaki. Mónica is currently professor at the School of Biological Science at San Marcos University and a researcher at the Museum of Natural History in Lima, Peru. She had her undergrad studies at San Marcos University and her graduate studies at the University of Texas with a master thesis and a PhD at the University of Florida in the United States. Her main interest is in the evolution of plant diversity, and her work has been mainly in cacti and other representatives of arid environments. Today she will be talking about hybridization in Haageocereus and other Trichocereeae. So Mónica, thank you for the opportunity to have your talk today, and time is yours. And please keep your mic off during her talk.
Thank you, Matthias, and thank you for the this initiative of the meeting for the seminars. As I was telling before, this is really an old work that I did during my PhD. And so what I am going to present is first some background information. Everything has been published already because it's needed to understand the work we did on hybridization. So for example, the design of the markers, some chromosomal work, etc. And I want to start thanking the people who helped me in Peru in the field, especially Dr. Carlos Ostolaza, who is always there for anything that I always needed since I was a student. And Blanca León, María Isabel La Torre, and Asunción Cano, Asunción, my former undergraduate advisor. And I was lucky to have them helping in the field when I needed to collect these specimens for this work. And my former PhD advisors, Doug and Pam Soltis, and Pablo Esperanza was a PhD student at that time, so he also helped me a lot, especially with discussions on all these new to me forms of reproduction in wild and cultivated plants. And so my interest at that time was tribe Trichocereeae, and Haageocereus was one of the most complicated genera. They are mostly erect. Many are prostrate, but they are considered columnar. Almost restricted to Peru, but they reach Chile, northern Chile. As you can see, there are many names, like 120 names for Haageocereus. Of course, they are not all accepted now, but more or less 72 recognized species at some point, plus 48 infraspecific taxa. But in 1999, the IOS reduced the number of recognized taxa to 21, and that was my hypothesis at that time. Those are the ones I recognized, plus 6 subspecies. But more or less in parallel, Natalia Calderon did a really great job on especially using morphology. I did the revision of Haageocereus, and she recognized 9 species and 6 subspecies. Here are some of the species of Haageocereus. Most of them have nocturnal flowers. They are white, some of them pink, red, And many of the flowers, many of them flower at night, but then the flowers stay open during the day. So bringing many opportunities for pollination by different pollinators. So part of this difficulty in Haageocereus is that, for example, you can see in the picture on the upper right, I can find this type of stems in the same population. Some of them with really clear spines or very dark, long, short, with bristles and hairs, or without bristles and hairs. So this is why there is this proliferation of names. There are many intermediate morphologies, and from that time I knew there must have been hybridization going on, and maybe genome doubling, but I didn't know because there were no chromosome counts. So I started with that. So I decided to do chromosome counts for as many plants I could collect, not only Haageocereus, but other Trichocereeae. And as has been reported for Cactaceae, the basic number is 11, and Haageocereus pseudomelanostele, the type species of the genus, is a diploid. But I also found triploids and at least 6 tetraploids, among them fulvous and multicolor spinous. And there are some morphological characteristics that later I learned can help me suspect if some are polygon, like a bluish color, reduced number of ribs. That's something I would like to discuss with Laura, maybe, if she has seen something like that. And among other Trichocereeae, I found tetraploids also in Cleistocactus. And Weber-Bauer-Cirrus was really interesting because there are 8 to 9 species recognized, and all of them are or tetraploid or octoploid. There are no diploids, no triploids, or hexaploids. And that made me suspect that maybe the origin of the genus is a hybrid, that has a hybrid origin. It's a sister, as opposed to what people believe. Weber-Bauer-Cirrus has several times been transferred to Haageocereus, but the sister is Cleistocactus. So if that, the idea that the origin is a hybridization, Cleistocactus can be one of the parental. And so this was published in Britannia, but one of the main objectives of the project was conservation, because most species of Haageocereus have very restricted distributions. In the extreme cases, only one population that is covering 2 square kilometers. Now, that was the case of 2 species. And only 2, I would say, have a wide distribution, considering wild just Lima plus maybe Ancash, or 2 or 3 departments. And so this is the place where we can find Haageocereus bacalaensis subspecies repens. And you can see that there is, at that time, several years ago, almost more than 20 years ago, when we collected this with Dr. Ostolaza and the help of students from Universidad de Trujillo, we moved many individuals to adjacent areas and to botanical institutions, also the university, because all that site is now covered with sugarcane plantations or asparagus plantations. So this is not longer there. No? And that's the only place where it used to be. And the other highly endangered is tenuis, and tenuis consists only of one population in the Department of Lima, and also has many problems because there are farms and also the city in Lima has grown until reaching these areas. So this population is also gone, but fortunately, people have been finding other small populations close to that area. But when we want to measure, in this case, the interest was measuring the genetic diversity in this. But for that, we need some point of comparison. So I chose the ones that have the widest distribution and supposedly no conservation problems. So what we will find in Haageocereus acranthus and Pseudomelanostele, that genetic diversity, we will consider the one that is average to high, and then we will be able to compare genetic diversity in the others that have conservation problems. So for that, we collected, it's interesting that when you find acranthus, usually you find Pseudomelanostele. And so we collected in these 3 populations, which are more or less separated by 50 kilometers. This is how acranthus looks in habitat. And Pseudomelanostele, which is really abundant in the places where you find it. And so now I'm going to talk a little bit about the markers used. So there are several markers that were available at that time to do this type of work, but we chose microsatellites. The problem is to obtain the microsatellites because those are designed for a specific genera in these cases that we did for Haageocereus, but sometimes they can be applied to other genera in Cactaceae, for example, or sometimes when there is higher variability, you can only use microsatellites for the species for which they were designed. They look like this. They are tandem repeats in the genome. They look like this. These are called perfect microsatellites. Here, there are 3 different microsatellites. This is a composed microsatellite, or some of them are interrupted. So they are tandem repeats. The advantages are that they are abundant and they are almost uniformly distributed in the genome. So you have good representation, especially in nuclear genome. Highly polymorphic. So the number of times the sequences are repeated will vary between individuals. And so we are able to distinguish one individual to the other. Codominance inheritance means that you can see the contribution of the parent, paternal and maternal. And the evaluation is relatively simple, it's through PCR and electrophoresis or fragment analysis. So once you amplify, we design the primers. That's what we call capture the microsatellite. So we design primers specifically for Haageocereus. And when you amplify these portions of DNA that have the repeated sequences, if you run a gel, you will see something like, like this. In homozygous, we'll have 2 copies of this. This is 1 individual, 2 individuals. This is the 3rd and 4th. In cases like this, when you have 1 band, that means that the maternal copy and the paternal copy are the same. And so that's a homozygous. When the maternal copy and the paternal copy are different, you have heterozygous. And heterozygosity can be interpreted as more diversity. That's what we are looking for, more heterozygous in the population. If this is when we evaluate through gel electrophoresis, but we use the same equipment that are used to obtain sequences, DNA sequences, can be used for fragment analysis. So instead of bands, we get these peaks. This is the short peak, the small peak, which is this, and this is the large, which is equivalent to this size. And then we can do the analysis. So this is what you expect in a diploid. And with those results, we first, I didn't mention that for the Y, the Haageocereus that have the Y distribution, we collected in 3 populations, and it's recommended to collect 30 to 32 individuals per population. And that's what we did. So for Pseudomelanostelia and Acaranthus, we have 30 to 32 individuals. And for Tenuis, we collected 45 individuals, and for Repens, 30 individuals. We ran an AMOVA with the results because we wanted to see if the populations were structured, if they were different, or they could be considered only one population. The 3 of them, only one population. And what we found, this goes from 0 to 1, and 1 will mean that the populations are completely different, one to the other, they don't share alleles. And 0 means that the 3 can be considered one population because there is exchange, genetic exchange. So a 0.04, that means very, almost no structure. That means that the 3 populations can be considered one. There is no difference among those populations. And we also found that the heterozygosity is really high. This also goes from 0 to 1. And so this is what you expect from a healthy population, is what we expected for Pseudomelanosteria. And the low levels of differentiation among the 3 populations were also expected, given that they are pollinated by bats, and bats, also hummingbirds, are known for traveling long distances. So these results are what we expected. But when we did this for Bacalaensis, we had still high levels of heterozygosity, which we didn't expect. And we interpret this as maybe there hasn't been time to see an effect on the genetic diversity of this species because the fragmentation is recent. But we also know from the literature that other rare species of cacta and also their woody plants of arid zones fix high heterozygosity. Sometimes they mix clonal reproduction with sexual reproduction. And that's something that has been observed in other plants. Now, tennis was a surprise. Remember, I mentioned there was only one population covering 2 square kilometers, and they look like this. So this is what I consider one individual, because the main way of propagation is stem fragmentation, and winds are very strong here that can move one branch. But microsatellites, when I ran the microsatellites, so I ran the microsatellites before counting the chromosomes, and when I saw the patterns of the 45 individuals, they were all identical. That means the whole population is one clone, a single clone. And there has been reports of clones in Stenocereus eruca in Mexico, but they found several clones, not only one, like in this case. And then I counted chromosomes immediately, because another thing that I saw was 1 to 3 peaks or bands. That means signs that there is a triploid. And a triploid, we expect from a triploid reproductive problems with reproduction. And counting the chromosomes, yeah, I found that it was really a triploid, and that examining the seeds and dissecting the seeds also could identify that they are the type of apomixis or reproduction, asexual reproduction by seeds, is by adventive embryony, like happens in oranges. I don't know if you have seen that. Because of the triploidy, the zygotic embryo, the zygote degenerates and is replaced by false embryos that have maternal origin. And sometimes not only one embryo, but several embryos. And sometimes they fuse and they produce seedlings, like in the picture here, that have more than 2 cotyledons. In this case, it's showing 3. So I found like 70% of the seedlings I obtained have 3 cotyledons. So now I do, when I germinate cacta, I look at this, and if I find that there are more than 2 cotyledons, I can suspect that there are probably apomixis happening. And so then, how we get a triploid? Usually, when we find hexaploids, the way that happens is that first the hybridization between a diploid and a tetraploid, and you get the triploid, and then doubling that triploid, you get an hexaploid. And we have found hexaploids in Espostoa. And so this was also published. We hypothesized who would be the possible parents of this triploid. So this is the place where we find tenuis. And the only diploid that is sharing the habitat with tenuis is Pseudomelanostelia, which is erect. Doesn't have this growing form. But other prostrated Haageocereus are towards the south. And we have multicoloris pinus, which is tetraploid. We have Chalaensis, which is tetraploid, and another one that has a wide distribution, which is decumbens, and is diploid. At that time, we didn't know that there were decumbens here, but we find decumbens until Lima. So probably decumbens could be the diploid parent, and one of these, the tetraploid parent. But we still don't know where this hybridization happened, how did it got here, or if we need to, well, we still need to explore more this area. But until now, this is our hypothesis of how we got this triploid clone. And reading in the literature about how they treat this type of, because can we consider it a species? It's in the red list of endangered species. Should we protect a clone? So those are questions that come when we are doing the categorization. And so I found that these are called microspecies. So the characteristics of microspecies is that they have a hybrid origin, a hybrid constitution, but they are distinguishable from other close relatives. And they have special requirements in habitat. They occupy a certain place. And so it fills all the characteristics that define it as a microspecies. And when I am asked about conservation, I will say that, yeah, we should protect this. This has, as I told you, the origin of an hexaploid usually is through triploidy first, no? So it has a evolutionary future. There can be mutations that will happen. Also, we can say that this happened not almost relatively recently, because we couldn't find any mutations in 45 individuals. But mutations will happen. Maybe it leaves the sterility, so it has evolutionary future, and we should protect it. Just so we can compare the number of alleles. So this is a summary of This is taken from the paper where we report the design of the microsatellite primers. So we designed 16. In most of this work, we have used from 5 to 11 microsatellites of the 16. Now that it's easier to obtain microsatellites, we are required to run, I don't know, 30, 40 or more microsatellites. But at that time, it wasn't that easy. So we only work with 5 to 11. And these are the 5 microsatellites. And these are the alleles that we found in the triploid. And compared to the number of alleles that we found in Pseudomelanostele. So we found 2 for this microsatellite in the clone, while in Pseudomelanostele we find 16 that range between 221 and 253 base pairs. We found 3 in the clone, we found 13 in populations of Pseudomelanostele. And so. Now, there are some limitations in the use of microsatellites. We found that Acranthus was tetraploid from chromosome counts, but also by using the microsatellite, you can see that. So in a tetraploid, you can find 1, 2, 4 alleles. And to find genetic differentiation or to explore if there is a structure or no, you need to know not only the allele number, but the allele frequency. And frequencies are difficult to calculate when you have this. Because if you have 1 peak or 1 band, that means that the 4 copies in a tetraploid are the same. And if you have 4, you know that you have 4 different copies, but you always need to have 4. When you have 2 or 3 is when you have problems finding the frequency, because here you don't know if there are 2 copies of this, or this, but one of these needs to have 2 copies in order to have the 4. In this case, you can have the alternatives are 3, 1, 1, 3, or 2, 2. So that's the limitation when using microsatellites with polyploids. But still, just given the number of alleles you find in the population is a measure of diversity. So here I place Acranthus, the tetraploid, Pseudomelanostele, the diploid. So these are the ones with the largest distribution. And repens, tenuis, and tenuis, the ones that have conservation problems, no? Because there is only 1 population of each. And so you can see that the tetraploid have highest genetic diversity than the diploid widespread. And this higher than the diploid that consists in 1 population, and the clone has only 2 alleles. Now, I can use the same markers. The idea of using microsatellites is because it has many applications. And so I use the same markers to examine hybridization. I just put this definition because even for me at the beginning it was difficult to find a definition. And I like this one, which is, and we want here to think more than in populations than thinking in species. So hybridization is defined as the crossing between 2 individuals that belong to populations or groups of populations that are distinguishable on the basis of 1 or more heritable characters. But most people recognize hybridization that occurs in natural settings, natural hybridization, because you know that in cultivation we can recreate many. But what happens in nature, but also the one, many are ephemeral, but the ones that we are interested in are the ones that produce progeny, and so there are evolutionary effects from hybrids that will pass this F1 generation. So we have parent A and parent B that produce an F1. The chances that an F1 will find another F1 and produce an F2 are extremely rare. So what usually will happen is that the hybrid will backcross to one of the parents that is more abundant in the population. And depending on with which it backcrosses, you will see the effect in the morphology. So at the beginning, you see an intermediate morphology. As it starts backcrossing with one of the parents, it starts resembling more that parent. And sometimes, like it happens to us, we don't distinguish the hybrid. We cannot tell apart which is the hybrid and which is the parent. So that it reverts to the form of the parent. This is from Rowley, and there are many hybrids that have been found later after 1994. But just to give you an example of how complicated it is working with Trichocereeae, because these are all Trichocereeae except for brown ninja. So it also hybridizes with brown ninja. And I know that there are similar cases in the Serie in Brazil. And it doesn't matter the pollination syndrome, because some of the diurnal flowers remain open until late hours of the afternoon, or some of the ones that open at night remain open during the day too. And so some of the ones that we find with more frequency are, in my case, the Haageocereus with Haageocereus, or Espostoa with Haageocereus, Echinopsis with Haageocereus. Also, from personal communication, I have heard of more hybrids between Haageocereus and Echinopsis. But the reason I took this is because I don't know of many hybrids here that are not ephemeral, that pass the F1 generation, except for this combination. And so there have been reported 7 hybrids between Espostoa and Haageocereus, but I was only able to find, well, I want to introduce Espostoa. Espostoa is very easy to recognize because they have this cephalium. Most of Espostoa have large amounts of hairs covering the stem, but especially in that area where the flowers and fruits emerge, and so this hair will protect the reproductive organs. We have most of the species in Peru, and we have 2 in Ecuador. Besides these, as I mentioned before, Natalia Calderon was working with Haageocereus too, and she also attributed several of these names, Haageocereus names, to Haageocereus, which is another name that was given to the hybrids, and she suggested that these have hybrid constitution. So these are the 3 combinations I was able to find. And so the problem is that these hybrids have been given general names like Bingamia, later transferred to Neobingamia, also Haageocereus, but now we are using this. This is the proper way of designing hybrid, the X and Haagespostoa. And this is one of the combinations. So in the hybrid, you will usually see that have the size of Espostoa, but the external appearance of Haageocereus. But you can see that there's a very big difference in size between the hybrid and Haageocereus. In a population that is dominated by Haageocereus, you find 1 or 2 Espostoas and maybe 1 hybrid, 1 or 2 hybrids. So that's what I was able to collect. I collected 32 individuals of Haageocereus, the hybrid, and the Espostoa. This is the second combination. And the hybrid kind of a cephalium, no? It's not like the cephalium in the Espostoa, but it's interrupted. And this is the third combination. So the first question we have is, who is the pollen donor? Considering that we have only one Espostoa, many Haageocereus, we hypothesize that the pollen will go from Haageocereus to Espostoa. And so we use chloroplast markers, which are maternally inherited in most cacta. And here we have the three hybrid combinations. So I have Espostoa, hybrid, Haageocereus, the same. And when we sequence the PSVA TRNH, this spacer, we didn't get differences in size, so we have to sequence. And what the sequence tells us is that it shares the chloroplast sequence with Espostoa. That means Espostoa is the maternal parent. And Haageocereus the pollen donor, as we hypothesized. And when we did two more markers, well, we sequenced two, but we don't. We can see the same results just by looking at the amplification size. Like in all cases, Espostoa and Haagespostoa share the sequence, and Haageocereus have a different sequence. So in all cases, Espostoa was the maternal donor. Now, running microsatellites. This is a way of presenting microsatellites suggested by Pablo. So when I put the arrow in this direction, that means that Haageocereus is the pollen donor. The positions of the samples are like Espostoa, hybrid, Haageocereus. What we're expecting on F1, we're expecting on F1 a complete correspondence of alleles. Es decir, the alleles in the F1, we were expecting the F1 having the alleles, the same alleles that have Espostoa, plus one of the individuals that, if we are lucky of sampling the one that was the pollen donor. And it happened with the second combination and the third combination. Should happen that the same. But what we found is that there is no complete correspondence of alleles between what we consider the hybrid and Espostoa or any of the individuals of Haageocereus. But of course, these alleles are present in the population. So that means that what we sample as an F1 is not an F1, but is a later generation hybrid, can be an F2 or a backcross. So the hybrid, the F1, must have dying. It's not present in the population anymore. The same happened with this combination. In this case, we only collected 3. And so we use more microsatellites, but the result is the same. So running another program, doing another analysis using a Bayesian approach that can find probabilities, posterior probabilities of each individual belonging to one of these categories, assigning it to pure parent A, pure parent A, Espostoa, pure parent B, Haageocereus, and F1 and F2, backcross with parent A, with backcross with parent B. And they are in the same position, Espostoa first, and second place, the hybrid, and then Haageocereus individuals. And what you can see is that several individuals that we identify as Haageocereus have higher probabilities of being backcrosses, no? Or backcrosses to one of the parents. And so those are characteristics that we find in hybrid swarms. So it's difficult now to say when we collect Haageocereus that we're collecting a pure Haageocereus. Maybe there is no pure Haageocereus anymore. So it's why it's so complicated with Haageocereus pseudomelanostellum. We couldn't do this work with this analysis with Acranthus. and I leave this again, all the hybrid possibilities. I would like to see if you have identified hybrids. What are your ideas about the hybrids you have seen? And I want to thank because this was part of the PhD work. At that time, I got funded from the American Society of Plant Taxonomy, the BSA, the Cactus and Succulent Society of America, the International Association for Plant Taxonomy, and the National Science Foundation. I want to thank also the Ministry of Agriculture, who always support us with making easy to obtain the permits easier. And so I leave this chart. Thanks.
Thank you, Mónica. Really nice work and tough. Say the least. And if you want someone have questions, please just can turn on your microphone and your video cam also to talk with Mónica. Tanya, go ahead.
Thank you, Mónica, for your presentation. Very interesting and a lot of work you have done. I have several questions, but I'm gonna do only a few. So in one of your first slides, you show plants in the same population have very different stem morphology. Same population. So do you have any idea or theory about why is this happening? Is there microclimatic differences? Because I have seen this in Mexican cacti, but not these dramatic differences.
In this case, I will say is because these are a process of hybridization, and some might be F1s, some might be back crosses, several generations of back crosses. And so each time they are resembling more one of the parents, and also it's polyploidy too. They are larger plants, more robust, and I have found that in Esposto, I have found diploids, tetra, and hexaploids. It's prevalent in some of these, and so I will see the polyploidy will give you, like I mentioned, I see a bluish color, I see a larger stems, sometimes different number of ribs, but things like this because of hybridization with Espostoa.
How complicated.
Yeah, and so all the different names, I don't think we can assign different names to this, and this will stay as a swarm, hybrid complex or hybrid swarm.
And given all this hybridization scenario for thinking in the evolution of these plants and elucidating the phylogeny, what method you think could be useful to elucidate the phylogeny? Because you can have hybrids here, even in between different genera.
Yeah, I mean, the good thing is that if you know the group, I think you are able to recognize it. And of course, you have to take them out of the phylogeny because it will really cause problems. But you can incorporate them just to show evidence that they are hybrids, and then you will have to take and show like this might be a hybrid between this and this. And there are other tests that you can run just to support this idea that you have that, because at the beginning it's just evidence from the phylogeny that you have a lower support when you incorporate it, or you have chloroplast markers that give you one result and then nuclear. And that was time ago. I asked how are you doing to reconcile these different results you are getting from transcriptomics, and because I'm getting constantly that type of problem. But I think there are ways now. There are many tools to identify these, and then we have to treat them differently. But I was surprised of one thing, that Acranthus and Pseudomelanostele, one is tetraploid and the other is diploid. These different employee are keeping them, they don't mix, they don't hybridize, and they are always living close, very close, but they don't hybridize. I haven't found hybrid. That was my first, I suspected that they will hybridize, but no. So that difference in chromosomes is keeping them separate. But another thing that I'm surprised of is, in the phylogeny, I get all the Espostoas monophyletic. Espostoas hybridize with many, but it's good support and everything. No problem with resolving Espostoa. It's interesting how Espostoa can hybridize with, and it's one thing I will ask Laura. What things can be done at that? But you see how just knowing the chromosome number helped me a lot at the beginning.
Yes.
Finding this triploid opened so many other questions, and see, yeah. So now those are some of the things I'm trying to do more, because before I didn't have the material. I wasn't in Peru. I was limited to the material I had for the phylogeny. But now that is why I started looking more into this, because I can go to the field.
And finally, are you doing any conservation actions for repens and the other, I think, was tenuis?
Yeah, well, Pacalaensis repens the population is gone. So, but I still have doubts.
You got to say few individuals you mentioned?
Yeah, we moved few individuals, but then I have doubts about this being a good subspecies. Maybe it's Pacalaensis, because the substrate is very different. In the place where repens was is almost prostrate, and it's sandy soil. Pacalaensis is in a loma, and it's a little more erect, but the difference is not large among them, at least. So Pacalaensis and tenuis, people have been finding tenuis around that area where it was described, but I'm doubting that it's tenuis. I think is one of the parents probably. And counting chromosomes on one of them is I have doubts about the ploidies, so I'm not sure if they are finding tenuis. Still, it's not in danger because we have the tenuis plant and it's a clone. So you have just one plant, you have all the diversity of tenuis. But it's good that they have found. I think the parent will be interesting. But several people have tenuis growing in cultivation. But yeah, it's sad because other Haageocereus are facing the same problem, and even larger. It's very difficult. I mean, at that time we could do something, but now it's really hard because these are growing where the largest cities in Peru are. These are coastal mainly.
Thank you, Mónica.
Thanks.
I've got a quick question, Mónica. What is the so the Haageocereus what is it, tenuis has all that trash around it? Did I miss that part?
Oh yeah, it's full of trash because there is a poultry. How do you say? Granjas de pollos, chicken farms.
Oh.
Chicken farms around. Yeah, around the area there are chicken farms, and a lot of, in most populations, the problem is that there is a lot of problem with the management of this trash, and they get trapped because of the spines and and there are strong winds, but this trash gets trapped by the plants.
Okay.
Paper is the main problem. Towel paper or plastic bags that are covering the stems. Not only of this, I'm talking about other coastal cacti.
Okay. And you were saying this population was destroyed?
Yes, there are not houses, but people have divided up, first flattened and then divided, this time to build house.
Okay.
Yeah.
I was also curious if you could explain one more time how you see apomixis by just looking at the cotyledons.
Not just by that. So,
oh, I see. Okay.
If the order was, I mean, if I counted the chromosomes and find a triploid, that's what I will. If I have seen that this is producing fruits and seeds, then I would say how it produces seeds if it's triploid. And during meiosis, there's problem pairing, so it shouldn't produce seeds.
Oh, okay.
It should abort the seeds. And say, but it's producing in cultivation, is producing seeds, and so. But I didn't get to count the chromosomes at that time, but I ran the microsatellites and I saw from 1 to 3 peaks. That means this is not diploid, this is triploid. And it's 45 individuals is a good number. So I could say it's triploid. But then we're all the same. So then I understood. So then I counted the chromosomes to confirm. I said that's the reproductive program it has. It's sterile. It's sterile, but it's producing seeds. So I had to examine the seeds. And there are several ways of reproducing. There are at least 2 apomixis types, and one is adventive embryony. And I dissected the seed and found several embryos, and also what I mentioned about the cotyledon. No? Where is it? So if you see in the literature for Opuntia, they mention 3 or 5 cotyledons. So this is not rare. In Opuntia has been reported. It has been reported in other cacti when embryos fuse and produce seedlings with more than 2 cotyledons. So now if you grow cacta and you see more than 2 cotyledons, it will be nice. It will be good examining if something like this is happening. But cacta mix, it's amazing how they can. This is perfect, and it's why agronomists, like I discuss a lot this with Pablo because he works with paspalum, and agronomists look for inducing plants to be apomictic because that's perfect if they produce asexual seeds, keeping all the characteristics of the desired characteristics. And seeds are easy to manage, and in the wild, seeds can be dispersed farther, and they are still clones. It's a very effective way of.
Awesome, thank you.
Teresa? Yes.
Yes, thanks, Matias. Hi, Mónica.
Hi. Hola, Teresa.
Hola. I wonder if you can give us an explanation why so many hybrids in this region. Is related to the abundance of certain species that are diploid, or no floral barriers are, so it is easy to cross. I was surprised that they look as one of the parents, and it's a lot of molecular data similar to the hybrid, and probably there are many other species in Cactaceae when populations are so high that have that problem. They have that abundance of hybrids and we haven't noticed it. Can you give us a little bit more of an explanation about this?
Yeah, thanks for your question, Teresa. So I don't know if you have seen phylogenies of Cactaceae, the ones we work with Erica Edwards. You see that the youngest group is Trichocereeae, and diversification of Trichocereeae probably happened after the formation of the Andes. So the high elevation in the Andes having reached like 5 million years ago, no? And that's pretty young. And in a group that is that young, speciation is still happening. And it's why I prefer to talk about populations and not the species, because they are in the process. And I think that's one of the reasons why Trichocereeae is like that. But remember that I'm here talking about only Haageocereus and Espostoa, and it's not in all genera of Trichocereeae. It's in some species and in some genera is more prominent. But Opuntia is another. I don't know how Lucas deals with that, but Opuntia is a larger problem than this. But in Trichocereeae, in the same way we saw polyploidy is not, because at the beginning I thought polyploidy is prominent in Trichocereeae, and it's not. It's in certain genera. Okay. And it's closely related. The ones that have more hybrids have more polyploids. But it's not happening in all. And another reason is that the pollinators, yeah, there are more generalists than we thought. Because these are places really harsh. I mean, we are in the driest desert. And in some places there is only cacta, no? And pollinators are scarce because, and so we see a lot of clonal propagation too, because pollinators are scarce here. But if pollinators are there are windows of opportunity, like during El Niño event, there's a humidity for several months. And it seems like at that time there's a renewal, recruitment and visitation of pollination and reproduction. So those small windows of opportunity are enough to introduce diversity. And then diversity is maintained by clonal propagation or other means. But it's, remember what I mentioned about woody plants, including cacta, in deserts have high genetic diversity when you expect poor or low. So they have other strategies to keep this diversity.
Thank you. Thank you, Mónica.
So Mónica, you bring up, Opuntia and hybridization and Opuntia. It's something definitely thought about a lot, why it happens so often. And, I think that one of the same things you bring up, the young age of the group, I think perhaps a bit to do with it. Presumably the tribe Opuntia is relatively young, right? And then it seems, and maybe this is, and maybe actually Laura could say something about this as well, but the apparent, more or less homogeneity of chromosomes in Cactaceae seems like it might help, ability to hybridize and maybe even potentially form viable hybrids more often than maybe you see in other groups of plants. But yeah, it's when you show the schematic of all the different hybrids among all of the different genera within this group, it is very, very striking. And it is very reminiscent of what you might see sort of across Opuntia. But I think one thing that's really impressive is you have so much intergeneric hybridization within this group.
I think you have found Consolea and Opuntia. You have found Consolea and Opuntia.
Yeah, that's correct. Yeah, we do have hybridization events between Consolea and Opuntia, and actually in the Cylindropuntii, we have one instance of hybridization between Grussonia and Cylindropuntia. So, but it doesn't seem to be quite as common, the intergeneric hybridization in those groups as it is in certain cactoids, which is really interesting.
Yeah, and just doing a survey, I found more common in columnar cacta in the wild, but more common in globular cacta in cultivation. Or maybe it's because the globular are more popular for cultivation.
Yeah, yeah. So I wonder, in terms of the columnar cacti, you know, is there any association with specific pollinators and the amount of hybridization, or does it seem to be?
Can be. I mean, they want to be seen.
Mhm.
They talking to what I forgot the name. From Mexico, who have published a lot about pollination in columnar cacta. I forgot the name, but I was talking to him once.
Alfonso Valiente?
Alfonso Valiente, with Alfonso Valiente. And he said, well, these columnar cacti have evolved in places where there were trees, no, like dry forests. They need to be seen. and now, if you go to Arequipa or southern Peru, you see lots of columnar cacti and no trees. No? And but they are mainly in Peru. These are mainly because Trichocereeae is diverse in Peru. In Brazil, they have the Cereeae, and they are finding similar things. Mostly pollinated by bats or hummingbirds, but the small ones by bees or others. But maybe. Mm-hmm.
But Haageocereus, that.
Haageocereus bats, the nocturnal bats or moths.
Moths too. So have you been able to like, I don't know, check some biological reproductive studies, like to check overlap between flowering periods and the overlap between pollinators and the preference between, I don't know, something like that?
There was one that I mentioned in the paper, a very detailed study by Catherine Salley, who stay in the field, and Haageocereus beverbaueri, who used to be 2 species and one of them with 4 subspecies, and they are all considered one now. She found that during years with El Nino event, the bats don't go and are replaced by hummingbirds. Is why there are 2 types of flowers maintaining the population. There are 2 types of flowers. You see tubular white flowers, and then you have a zygomorphic wine reddish color. The 2 are in the same population. and I think in other plants there is very slightly the same example I have found in salvia and other work I've been reviewing, and where they find the same, the 2 different pollinators, but then the difference in the flower shape is very subtle, but it's important for avoiding one pollinator. But then there is Carlos Martel. I don't know if you have heard about him. He studies essences, like essences, and he have found some in cacta. He mentioned that I mean the morphology is a bad syndrome, but the essence is not for bad. So now the cacta, as I mentioned, are more generalist, and I don't know what will happen in. I mean, it's a lot of investment in that big flower. No? Maybe cacta will start reducing the flower. No? Like San Pedro has a really huge flower, and it stays open 1 night, 2 nights only. And if it's not visited by a bat, but pollinated by a bee, I don't know. There's a lot of resources going to that flower. I don't know what will happen with that, but it's something that several people are interested in looking at. Mm-hmm.
Okay, we have more questions. Really nice, Mónica. I really enjoy it.
Thank you.
I feel better because I was not completely aware because I'm working with Opuntia and I'm facing similar, but I'm glad that I'm not alone.
Yeah, I know Opuntia is really complicated. But so I don't know if Laura is there.
Yes.
Yeah, I think it would be nice if you can do some fish or that type of work with the Opuntias. I mean, I wish I can do some of that work to really prove what we think is happening is happening.
Yeah, the parents.
I think that the cytological work cannot be replaced by molecular. Mhm. Thank you.
Thank you, Mónica. Really, really nice talk.
Okay.
Mónica, acá, perdón, acá estoy en dos salas diferentes. Te escuché, Mónica, sobre sí, lo interesante que sería hacerlo de fitogenética. Sí, tendríamos que, me parece súper interesante esto del triploide, ahí estaba viendo los cromosomas que existen y me parecen súper interesantes. Muchas veces uno cuando publica los triploides, por ahí,
me gustaría ver.
Ese es súper interesante, sí. Porque bueno, a nivel molecular, va a ver también qué porcentaje de genomas tiene de cada uno de los parentales. Sí, eso sería súper interesante, sí.
Sí, conversamos luego.
Sí, después conversamos, sí.
Sí.
Yo puedo ir a Perú, no tengo ningún problema.
Sí. Bueno, y después entonces conversamos un poco sobre, hemos estado haciendo bastante trabajo de campo en últimos 2 meses, pero creo que luego trataré de conectarme en la reunión que van a tener y conversamos.
Bueno, hermosa la presentación, muy linda, me gustó mucho.
Gracias.
Y Mónica, hay también nuevos métodos como de network que puedes calcular la probabilidad de que haya el flujo y los eventos de hibridación, ¿sí?
Sí, ahí sí necesito ayuda todavía.
Dale.
Sí.
Ok.
Sí, muchas gracias entonces.
Gracias, Mónica. Gracias a todos. Bye bye. See you soon and keep tuning.
Sí. Sí.